All right, so welcome everyone. Today we're really excited to have Raphael speaking today. And today he'll be discussing native metabolomics for the discovery of new protein modulators. So excited to hear about that and the progress he's been making now recently. But before we dive into the science, I'd just like to give a little background introduction to Raphael. So he started studying pharmacy at the University of Münster, and he actually obtained a license as a pharmacist from there. So I recently learned that. But then during an advanced lab course there, he was first introduced into the world of natural product isolation and structure elucidation. And I think that really captured his fancy. So then he moved to Bonn, where he received his master's in drug research. And that's where he wrote his master thesis, which actually focused on natural products from symbiotic bacteria of Chinese medicinal plants. From there, then he stayed on to receive his PhD, where he was doing chemical, pharmacological, and ecological aspects, looking at different specialized metabolites. Then he was funded by an NIH grant to join the Gerwick Lab as a postdoc at the Scripps Institute of Oceanography and Skaggs School of Pharmacy at the University of California in San Diego. And that's where, well, Daniel, him and I all met. And that's also where he developed some new NMR and mass spec-based tools towards the automated structure elucidation from natural products produced by cyanobacteria. I think he might be talking about some of that today, but I'm not entirely sure. After that, though, he moved and he started exploring cryo-EM and micro-ED for structure elucidation of small molecules from mixtures. And then recently, in March 2022, he was actually appointed as the professor of pharmaceutical bioanalytics and natural product discovery at the University of Marburg. And that is where he's coming to speak with us today from. So yeah, take it away.
Oh, thank you so much for this very detailed introduction. So I think I can skip mine. That's perfect. Thanks a lot, Allegra. And yeah, it's my honor and pleasure to talk in this VMOL series. And it's now kind of a follow-up on what we heard from Allegra last week. So I will tell you about native metabolomics for the discovery of new protein modulators. Just so that all of you who are not natural product chemists get an insight. So natural products are also referred to as secondary or specialized metabolites, and they're produced from all kingdoms of life, producing various bioactive and very interesting chemical structures, which have been used for more than 200 years now for drug discovery purposes. But they can also be very toxic and even have dramatic impacts on whole environments. One certain project I really like, because it's also from my former PI in Halle, Professor Niedermeyer, who worked with his US collaborations partner on a mysterious phenomenon where the bald eagle was killed or died in large numbers, and that happened to co-occur with an invasive plant here that you can see here, Hydrilla verticola. And then humans try to get rid of this plant and put in herbicides containing a lot of bromine, and a very talented cyanobacterium then produced this very potent toxin, a toctonol toxin, which bioaccumulated in the food chain. And so the ducks and coots that you can see here are heavily neurologically affected, and thus they're easy prey for the bald eagles, will then also die from this fatal neurological disease. Another more recent example is a local one from the Oder River catastrophe here in Germany. It's on the eastern border into Poland, where it also came to a massive dying event, in this case, a massive dying event of fish. And one major causative agent was this unicellular eukaryotic golden algae called Primnasium parvum, which produced these beautiful polyether leather toxins. So natural products can be important as drug leads, but they can also be very toxic and even shift whole environments. But they can also be used as pharmacological tools. And one tool I studied a lot in my dissertation work was this cyclic deoxypeptide we can see here, which specifically inhibits the GPCR signaling cascade. And we will hear about this more later on. But what fascinates me is that it is a non-ribosomal peptide supposedly produced by a plant, a traditional Chinese medicine plant. But if you have a closer look in this leaf nodules, you can actually see that those are filled with rod-shaped bacteria. And we speculated and we confirmed in the end that these bacteria are the actual producers of this compound. And the first experiment that we did was multi-imaging to show the occurrence of the bacteria in these leaf nodules here in the leaf rim. And yeah, so multi-imaging is a great tool where you can visualize or associate single molecules to organisms. But what always fascinates me is that even like a single bacterial cell, if you have a deeper analysis, you can see they are filled. Yeah, there's a new world opening, a new universe of molecules. And Allegra already pointed towards it. During our postdoctoral times, we studied metabolomes from various angles. So in the Gerwig lab, we tried to do metabolome investigations from the NMR side. And Nuno Banderas and Peter Dorstein's lab, the molecular networking method was developed. And in also many other labs in, for example, the Medeman lab or Tillman Weber's lab. There's also approaches to study DNA sequences to make predictions for the biosynthetic gene clusters and the structures that are expressed thereof. And in more recent years, the community tries also to find more interactions between these single research fields to unite them. And so those tools that are constantly developed by the whole scientific community, we want to apply and further develop to answer interesting questions in the field of chemical ecology, but also pharmacology, to get a holistic perspective on natural product structure elucidation and to see natural products of interest from various angles. So the tools I just showed you, the main reason for them is that we need them is, I showed you this multiverse of molecules. So we need methods that help us to prioritize and also to help us with their replication. So the avoiding redetection of already known natural products. And a powerful tool to do that are, for example, molecular networking. And if we have interesting compound of interest, we have to determine the structure. And that's also a common bottleneck, which can take, on average, I would say months for a single component. And therefore, we developed a smart NMR technique to accelerate the structure elucidation there. And only recently, we now try to apply and develop methods that are MS-based to do it all in once, to prioritize, to dereplicate, and also to annotate molecular networks. And that is only possible because of MassQL developed by Mingsheng Wang, at that time a postdoc in the Dorstein lab, now professor at UCR. And we used MassQL to investigate cyclic tetrapeptides we are interested in. And so we had a molecular network and we got MS2 reference libraries matches for 2 or for 3 compounds. But we wanted to know deeper the amino acid composition of all the nodes in this molecular family. That's why we wrote MySQL queries for all amino adducts of the amino acids, and that way we could do a substructure annotated molecular network. For example, some of those included proline, which was new, so we targeted them for isolation, and my PhD student Tim then was able to elucidate the structure and also to determine bioactivities. And so, yeah, we really like those tools to accelerate natural product discovery. But next, also important topic for me as pharmacist, but also as person who is interested in pharmacological or chemical ecology, is to assign earlier bioactivities to single components. And that's why I'm very happy that I met Daniel Petras, and who said, oh, I have a great idea actually. So let's maybe connect structure elucidation and direct bioactivity determination from complex extracts. And not necessarily the order how it happened, but it is nice for the storyline. We first had to query the current literature about what is available right now. So we are both from the field of metabolome analysis, but we also are able to characterize single metabolites. And additionally, Daniel is also kind of expert for proteomics, which is great. So we were looking for MS-based tools that enabled us to screen single protein of interest against whole metabolomes, which was done, led by Paolo and Amira. And so we found many great papers for offline approaches to search single proteins against metabolites. But as I just said, they're all offline. So we were thinking about developing an online approach to do that. And how we did that, I will tell you in the following. So we have first a crude complex natural product extract, which is separated by UHPLC. You may have seen these slides before because they are from Daniel, I think. So the mixtures are separated into single eluting components, and then with a makeup tube, the pH is adjusted to the pH optimum of a target protein of interest. And those are continuously infused and allowed to interact with the single eluting molecules. And then we acquire mass spec data. And so as a proof of concept experiment that we still started in our times during our postdoc, we used chymotrypsin and screened it against Rivolaria, a cyanobacterial extract. And what you can see here is now a lot of new peaks that are popping up, which are potential new chymotrypsin binders. And we were kind of intrigued by the sheer number of small molecules that we could detect by only analyzing one crude extract. So you can see in this correlation molecular network the target protein of interest. And then in the dashed lines, you can see the potential binders that we detected by native metabolomics. And then the diamonds here represent molecules that are actually isolated and elucidated the structures. So we also used our tools like Smart GNPS, but also Sirius from the Böker Lab and Canopus for substructure prediction, which helped us with the structure elucidation process. And obviously, we also determined the relative and absolute configurations of those molecules. But very importantly, we next had to validate that our binding assays are really also translated in a biological effect. And that was done by an orthogonal bioassay from the O'Donoghue lab, located at Skek's School of Pharmacy in UC San Diego. And we were very pleased that he confirmed that all of our newly isolated compounds were nanomolar chymotrypsin and also other serine protease inhibitors. But of course, we had to optimize our conditions. And so we did various experiments to optimize our screenings. And these optimization steps are only for this chymotrypsin. So for example, we did a titration experiment where we saw a concentration dependent increase of the ratio of the ligand protein complex in comparison to the apoprotein. We also investigated the influence of the pH on this peak area of this peak here. And as I told you, for example, chymotrypsin also has a pH optimum, and if it's too low, the enzyme is not functional anymore. So that's why we have the makeup flow. Another different or important investigation was to detect or determine the limit of detection. In this case, it was 1 to 10 nanograms on column. So if you think about it, I think we mainly used 1 milligram per milliliter crude extracts, and we injected 2 microliters of it on the column. And then the single eluting compounds must be abundant in amounts like 1 to 10 nanograms to be detected, at least for this chymotrypsin system. But I think that's great results that we could see detection over a range of minimum 3 orders of magnitudes. So if a compound was there in 10,000 times excess, we could still detect it and the other way around. Then we also had a deeper look on the makeup flow composition. So ammonium acetate is the standard solvent that you use for creating native-like positions that allows protein to be in their native folding. And we could not see any detrimental effect on the peak areas using various concentration. But what we could see is with increasing ammonium acetate concentration, you see these asterisks here. There is a elevated background noise due to ammonium acetate clusters in the lower M/Z range. So one conclusion is that the ideal ammonium acetate concentration is protein specific, and also that high ammonium acetate concentration can lead to formation of increased noise. So it's maybe better to analyze the higher M/Z values. Next, we also investigated the influence of changing MS parameters on the protein binder complex peak area. So for one, we investigated the EC source, so the temperature and also the S-lens RF level, where, like I would say, normal range changes did not have a big impact on the relative abundance of the chymotrypsin molassamide peak area. But if you use too high CIDs in the collision cells, too high collision energies, you have a drastic drop. But I would say like 60 to 80 EV is not what you would normally use. You would probably start with 20 or 30 EV. Then also, crucial for our approach is that you determine the influence of the final acetonitrile concentrations on enzyme activity. So that means, we have in our separation, we have the LC gradient, which goes up to 99% acetonitrile, but then we have the makeup flow to dilute it. And in our case, the maximum concentration before reaching the mass spectrometer was 33%, because that's the highest concentration where we still had a good assay window for determining the binding. Next, we also did positive and negative control experiments. So especially the negative control experiments were important for us, where we also used several PAINs, so pan-assay interference components. Many of them are polyphenols and also molecules with kinone moieties. And maybe if you have a look in the literature, you also see the beneficial effects of polyphenols. And you cannot really say that it's really the compounds. Sometimes it's the bad assays, because many assay systems react to phenolic groups, and then you have false positives. So to exclude this, we also screened these molecules. And last but not least, we also compared native metabolomics with direct infusion. When we infused via the crude extract via flow injection, we couldn't really see any XICs or EICs. You can see maybe some colors down here, but actually we needed longer pre-incubation. So if we pre-incubated chymotrypsin with the crude extracts for 30 minutes and then analyzed it, we actually did see binders, but we could not detect 30, but in this case 3. When looking at this right now, Daniel, I feel like maybe we should have a look into this compound again, because we haven't elucidated it, and it seems to have, in the direct infusion experiments, a pretty strong signal, despite being not very abundant in the sample. So maybe it's something we could have a follow-up on. So it's always good to have a relook on your data. And obviously, I would not be talking today if we could only do it with chymotrypsin. So we also did proof of concept experiments with various proteins of different size and shape, and some of them were monomeric, some of them were trimeric, and so on. And the biggest one, I think, to that date was around 80 kilodaltons in size. And yeah, I hope this kind of proved that we could develop a workflow that may accelerate future drug discovery efforts. And also, it finally allowed me to go back to my beloved PhD project, which is finding new G protein modulators. And now I finally also tell you more about what those are actually. So, so the project Native Metabolomics for the Discovery of New GX Protein Modulators is from my PhD student Amira, and is done in close collaboration also with the Functional Metabolomics Lab from Daniel Petras. So what are heterotrimeric G proteins? So first, what you have to know is that they are heterotrimeric G proteins consisting of an alpha that you can see here in orange and a beta gamma dimeric subunit that you see in yellow and green. And they are very important for humans and actually all metazoan organisms. In our case, for just to name a few examples, for vision, for blood pressure regulation, muscle contraction. So you need them to breathe, you need them to let your heart beat harder, faster, and so on. And they are kind of molecular switches. And the active form binds GTP, guanosine triphosphate. So 3 phosphate groups here. And then it has this conformation. And then it is an intrinsic GTPase. That means if it's active, after some time, it inactivates itself by cleaving off 1 phosphate group. And then this small change actually leads to a dramatic change in the conformation of the whole peptide. And it leads to the reassembly of the heterotrimeric G protein in the inactive state, which then reassociates with a receptor. Okay, what I'm talking about receptor, here we can see the beta-2 adrenoreceptor. And if now, for example, the natural ligand adrenaline or epinephrine binds to its receptor, it leads to a conformational change here in this G protein. The G protein releases GDP and GDP goes in, and that pushes away the beta-gamma subunit, and you have pleiotropic physiological effects, such as, for example, a muscle relaxation. And the first one who was able to crystallize this structure was Brian Kobilka. I think he was awarded Nobel Prize in 2012 together with Robert Lefkowitz. And so I don't think I have to highlight again how important G-protein coupled receptors are for our human body. So more than 1/3 of all prescription medication have them as target. But for me, it was very interesting that while we have 4% of our human genome encoding for these receptors, we only have 4 different proteins inside the cell that help to transduce the signal further. And there's not many known inhibitors. I think most of us are probably, if you have a look in your vaccination certificate, you will see pertussis toxin in there. Preventing you for getting pertussis, which is a very potent G-alpha-I inhibitor, but it's a very big protein. So it's not a nice tool to work with in the lab. And this compound I already showed you in the introduction here, the cyclic depsipeptide we call FR, is now the first small molecule inhibitor of this G-alpha-Q protein. So no matter how many epinephrine you inject yourself with, nothing will happen if you have this compound. So it's a very important tool to study complex biological processes. And also, I don't talk about it today. It is interesting for new strategies for drug development. We can have a discussion later if you're interested in it. But yeah, just as the quick recap, how we want to analyze compounds, we are very interested in how nature makes molecules, how it affects this function of cells and animals, and also why nature makes molecules like this. And in this case, for the mode of action, it binds here into this G-alpha subunit, and this prevents that the helical domain cannot, it's like a hinge, and it cannot open up. And so GDP cannot be released for GTP, so it always remains in the inactive state. And yeah, we did a lot of work in finding new analogs of it, also applying molecular networking and more recently feature-based molecular networking to derive the pharmacophore of the molecule. And also in the ecological sense, I showed you the multi-imaging beforehand. We are now pretty certain that the bacteria produces this compound for its host, which is the plant, to prevent it to be fed on by insects, but also mammals. So there has been a lot of work by many, many people, not me, it was a whole research consortium. And why I'm telling you it again, because I'm still not very convinced on this part, on the ecological role. I think there's still a lot to do, because if you do a deeper research, you find very similar structures in ubiquitous environments. You find it as obligate endosymbiont, as I showed you, but also in free-living soil bacteria. And for me, very interesting also in the marine environment, in this tunicates. And my medium-term research object, one of that would be how important is G protein signaling in the marine environment. And now I've let you waited enough. I finally, with the native metabolomics approach, we have tools to find new modulators for this G proteins. So I just briefly want to show it. I think it's better to have this in the discussion afterwards to discuss the setup in details. I just want you to have a quick glance on the setup in Tübingen from Professor Petras, who now moved to UC Riverside. And I think there will be a similar setup soon too. And so what did we do to change the conditions to make it suitable to work also for G proteins? So we had to further adjust the final acetonitrile concentration. So we played with lower microflow condition in the LC part. We also tried to adjust the makeup flow with a makeup pump. And because these proteins are, in comparison to chymotrypsin, very hard to get and very hard to make, we also urgently had to work on decreasing needed protein concentrations and also to have it stable. And I don't know if Daniel showed it before. Some proteins are also thermolabile. So Daniel is not only a analytical chemist, but seems also to be an engineer. So he made a custom-made cooler out of a computer cooler, and I think some magic with his 3D printer. And yeah, so now those are the first results from G protein measurements. So the first question we had to answer is, can we detect heterotrimeric G proteins? For this, we had to get G proteins, which were kindly made by Jonas from the Paul Scherrer Institute in Gebhard Schertler's laboratory in Switzerland. And more recently, also Dr. Daniel Hilger, who learned in Brian Kobilka's lab and who also is teaching now my PhD student Amira how to express G proteins in E. coli and some of them also in insect cells, which is not super trivial. And so we were quite happy that we could see for different G protein families, we could see the APO G alpha unit, we could see the GDP bound alpha unit, and if we screened the heterotrimer, we could also detect the beta gamma unit. However, we could not detect the intact heterotrimeric G protein. But for our screenings, only this alpha unit is of importance. And next, we also did proof of concept experiments regarding positive and negative control. As a positive control, that's why I told you so much about it. We use this GQ protein and a GQ protein inhibitor, this FR compound. And we first used the standard, so a purified FR. And then at a retention time where it does not elute, we only see the G alpha protein and the beta gamma subunit. And then at the time where FR elutes at 8.8 minutes, we can see a huge additional peak here and actually also see a slight decrease here in the GDP bound form. This was very encouraging. And then we did the first real sample. So we used the plant extract where the natural FR occurs in. And even though it's tiny, we could clearly detect the compound here too, and it was calculated to be there in 2.5 nanograms, which again kind of confirms the formally established detection limit of about 1 to 10 nanograms on column. Then we also did the negative control, so we used another G protein family here, right? And So here we didn't observe any mass shifts at the retention time where the FR compound eluted. Oops, sorry, I didn't skip that. So then we could finally do our first experiments. And so our expert for marine fungi in our lab is Tim. And we intended to screen marine fungal extracts against G-alpha-I proteins because there's a high sequence homology between fungal G protein class 1 and the human G-alpha-I proteins. And so that's our workflow right now in our lab. So we cultivate our fungal using an osmic approach. Then we get our crude extracts. Then we acquire LC-MS data and use it, for example, for molecular networking analysis, and also use it for native metabolomic screening. Based on the results, we detect the most promising hits, which are then targeted for isolation and structure elucidation. And obviously, we want then to confirm the bioactivity with bioassays. So here you see the TICs of selected marine fungi. And here is the results of one strain overlaid. So in green, you can see the native MS run here of the continuously infused G protein. And in gray below, you see the metabolomics run. And yeah, important also, the blue dashed line here is the total or the post-makeup acetonitrile concentration, which was never higher than 35%. Otherwise, the G protein signal would not be stable anymore. So we screened several extracts and, then investigated first hits. We saw some drops here in the TIC of the native metabolomics runs. And if we had a look here, we saw a first mass shift occurring. And what we then do is we take the delta MZ, so we subtract the observed mass for the protein binder complex minus the apo protein. And then we have to multiply it with the charge state. And then we get the approximate molecular or exact mass of the molecule. And then we do the same experiment, but without infusing the protein, so a pure metabolomics run. And then we can correlate the retention times here. And then we see also this mass 446 and actually another one. And yeah, I cannot tell you the structures as of now, but that's how it is right now. So we were also wondering, so we isolated our first hit components, this 446 mass, and we also found that this compound I showed you before would bind to this G protein. So what we did was we mixed this in an equimolar concentration. They both have also very similar masses, so it's a little bit difficult. And so we can see mass shifts. And if we now in the collision cell, we can apply HDD ramping, so we can ramp the collision energy. And what we wanted to do is if we see relative binding affinities, and I think what you kind of see quite well if you follow here, that this 446 compound at what is it, HDD 15 almost already is disappeared, while the endolite E complex is more stable. So that was kind of interesting that this endolite E tends to have at least based on this MS assay, stronger or higher affinity to G-alpha-I than M/Z. Now, again, a very important question because I see it often also strangely in the literature that people always assume that binding affinity also translates to a functional effect, which is not always the case. But we will come back to that in a very second. So the second follow-up experiment we want to do is now we test the same compounds, the purified compounds, with other G proteins to test the selectivity towards only this G alpha I. So we, for example, also tested G alpha S proteins and incubated with the ligands in 10 times excess. And for the 446, we did not see any mass shift. So we had selective binding to G alpha I, even though it was not so affine to the protein. But endolyte E unselectively bound to all other G proteins that we tested. And now this is, I would say, not worth nothing, but we had to validate our preliminary findings again. So we need an orthogonal bioassay. And this was also performed by Dr. Daniel Hilger, who did a GTPase turnover assay, which is kind of a cool assay because you have, again, this is the G protein, and I told you before, it's an intrinsic GTPase. So if it's activated, it cleaves itself with phosphate groups here, and then you have the inactive GDP. And what you can do now is adding a reagent which converts in a quantitative manner GTP to ATP. And ATP, yeah, it is known that with luciferin and luciferase, you can convert into luminescence. So you have light, a light readout. So what does it mean? So if you have an inhibitor of this GTPase function, for example, more GTP remains, meaning you have more ATP, meaning you have more light. And the opposite is true for supposedly activators. So for this endolyte E compound, where we saw in our assays that it unselectively bound to all G proteins, we could not detect any functional effect. But for this 446 compound in a concentration-dependent manner, we saw that it inhibits GTP turnover. So that was quite exciting for us. And we confirmed it in many other assays. We also confirmed it with the purified protein or just the alpha subunit. We confirmed it with a beta gamma unit 2, even though the potency is not great, we have to say, but it seems to be selective and functional inhibitor. And as I see, I'm running out of time. Very quickly towards another puzzle, which we haven't solved yet, but we were very intrigued that the same fungus also seemed to produce a G alpha I activator. But this was produced in very minor amounts, so we could only test a fraction so far. So you cannot see it. So it's almost invisible. This little red hubble here, I don't know. This is the peak we were looking for. And so Tim then also did canopus analysis to have at least some structural insights to the molecule. And it seems to be a cyclic peptide containing phenylalanine and leucine as building blocks. And I think he had a great idea. So he fed to the fungus leucine and phenylalanine. And what we hope for is to get an increased production of the 814. Instead, a new peak eluting at the same time appeared, which seemed to be structurally very closely related to the 814. So we hope now that we can isolate this one in sufficient quantities for structure elucidation and bioactivity, and maybe then find other ways to get to the 814. I spare you the details here. This is also what I would love to discuss, because we're very happy that Amira now was able to set up with Dr. Uwe Linnen, our MS core facility, our own native metabolomics setup, not as fancy as the one in Tübingen, but it works. And what is great, for the very first time, we can detect the intact heterotrimer. So we can also see the apoprotein, the beta gamma protein, but for the first time also the heterotrimeric G protein. And yeah, we analyzed it further. We deconvoluted the spectra with UniDEC. And we also saw for the first time this peak here at 87 kilodaltons, and could clearly assign the single building blocks here, alpha, beta, gamma, and the heterotrimeric G protein here. And just to be sure, we also did another measurement at another fancy MS device, the Synapta Waters device, and also could determine the mass for the heterotrimer. So concluding, we could detect and test all G proteins that we tested so far over the whole gradient against hundreds of natural products in only 30 minutes. And we already have identified several putative hits for G-alpha I binding from marine fungal crude extracts. I think what's so great about this approach is the versatility. So we can simultaneously also screen for alpha, but if we were interested in it, we could also look for beta-1, gamma-2 binders as a direct readout. And furthermore, we also get mechanistic insight. So I showed you this on state when GTP is bound, or this off state when GDP is bound. So we could also use the method to find state selective inhibitors, which is awesome too, I think. And we first had some hands-on experiments on trying to determine relative binding affinities using HGD ramping. I don't tell you about other projects we are working on right now. I really want to thank our slowly but steadily growing lab, and obviously all collaboration partners and also mentors from the past, especially also Daniel Petras and Daniel Hilger. And with that, I thank you very much for your kind attention, and I'm happy to answer your question, or we can also have discussions on the setup. Thank you very much.
Thank you so much. That was really cool. So I guess I can moderate questions then, but before we dive into questions, if anyone has to leave, yeah, I would say now is your chance. We'll stop the recording and then we'll take a few questions.